My name is Janine Lomar Kelly, and I'm a technical information scientist here at the Jackson Laboratory. And today I'll be moderating this webinar, Aligning Health Surveillance and Risk in Embryo Transfer. So let me go through a little bit of background about the Jackson Laboratory mission in case you're unfamiliar with who we are. So we're actually an NIH-funded institute tasked with the mission to improve human health and disease by understanding and using mice as disease models. And so we actually have furthered this mission in 3 different ways by performing research. We've got a number of investigators over in our main Bar Harbor campus that perform research using the mouse as a model for human disease. We also provide resources, including JAX mice in clinical research services. We also have a number of bioinformatics databases and technical publications to basically help serve you and your research goals as best we can. So you probably know us mostly from the mice that we provide. And then finally, we educate scientists using any methods of courses or internships and other programs for high school students all the way up until postdoctoral training programs. Probably many of you know us for our mice. We are the JAX mice, or the gold standard for biomedical research. What many people don't know is that we're an NIH-funded resource to collect and curate as many unique mouse genetic models that we can find in the literature. And so currently we have over 7,000 strains, and we're growing in that and accumulating and collecting more of these strains annually. Today's presentation is actually very relevant because we do accept hundreds of new strains every year. They're coming from a lot of different laboratories throughout the world. It's actually very important to be able to monitor what types of pathogens and prevent those pathogens from entering into our facility, and so that they're not passed on to anybody else in our facility or to anybody that we distribute mice to. Our mice are unsurpassed in genetic quality and animal health. We have very rigorous health monitoring, as we'll talk about today, as well as genetic quality monitoring and quality control. And the JAX mice are the best characterized and most referenced. Over 100 new publications per week come out using our mice. So you know that you're using mice that have a lot of research and background behind them. And what a lot of people don't know is that the common inbred strains, such as C57 Black 6, BALB-CJ, and DBA, which you can buy almost anywhere, the ones that you purchase from us actually directly support the development and collection of our specialty strains, as well as other valuable community research resources that I'll talk about next. Some of those resources are our JAX mice database, and this is basically a one-stop location to learn all about the mouse that you've purchased or a mouse that you're going to purchase. It's like a very extensive product catalog, and you can get links to breeding information, genotyping, references, relevant literature references, pricing, the development of the strain. It's really, I go to this page about 1,000 times a day. We also have the Mouse Genome Informatics database, which is a very gene-centric database to learn a lot more about the types of mutations that are present in any kind of mutant mice that you might be interested in using for your research. So this is a great place to come if you're looking for a new model or looking to see if there are any models that exist with a gene mutated that you're interested in. And then finally, the Mouse Phenome database is also a great database to learn a little bit about some of the basic baseline data from a lot of inbred strains. And so a lot of people will come here to look for clinical chemistry or body weight and a number of other kind of resources. This is actually a user-submitted database where other researchers can submit their data into our repository or database repository, and then it's freely available to anyone else to download and manipulate that data and graph it so that they can choose different strains, and also to verify that what they're seeing is also being seen by other people. And there are many other unique resources, so if you're interested in any of those, be sure to contact us in technical information, and we can probably help you find something that will help you expedite your research. Getting into our formal part of the presentation, that title is Aligning Health Surveillance and Risk in Embryo Transfer, and I'd like to now introduce our speaker, Dr. Rob Taft, who is the Director of Reproductive Technologies and Business Development here at the Jackson Laboratory. Rob earned his PhD in reproductive physiology from West Virginia University in 1999 before coming to the Jackson Laboratory to pursue postdoctoral training. Today, Rob's focus is on the development and application of new technologies for creating and managing mouse models. His efforts have included work on methods to reduce the rate of genetic drift, improved cryopreservation and in vitro fertilization methods, and improved host embryos for ES cell microinjection. And he's joined today by Dr. Jim Fahey. He's the Chief of Diagnostic Services and Associate Director of Lab Animal Health Services here at the Jackson Laboratory. And before coming to JAX, Dr. Fahey was employed in the biologics industry working on vaccine development for human and animal infectious diseases. Dr. Fahey did his postdoctoral studies at the Trudeau Institute, a renowned research institution for the study of immune response to cancer and infectious diseases. And since graduating from veterinary college at Cornell University, Dr. Fahey became board certified in veterinary microbiology. So as you can see, we have some great expertise here in the topic about health surveillance and embryo transfer. And with that, I will introduce Rob to take it over from here.
Thank you, Janine. Can you hear me okay?
Yep, you sound great.
Okay, great. Thank you. First off, I'd like to thank everyone for attending the webinar today. We're really happy to have the opportunity to present this to you. It's the outcome of some work that Jim and I have been doing collaboratively for the last couple of years. And I think it exemplifies the JAC's commitment to animal health and quality. A key objective for us is to produce the healthiest, most genetically stable and well-defined research mice possible. And I've been here now for about 15 years, and I can say from my experience, this is something that really permeates everything we do. And that begins with the facilities, the equipment, the PPE that's used every day to help ensure that animals remain of a very high health status. It's an integral part of all of the processes that we use, and it's a key element in the training that we provide. Every new employee that's going to be working in animal care spends weeks in training, becoming proficient in all of the methods, and this idea of that we really have to produce the healthiest and highest quality mice possible, something that's really emphasized during that training. And then of course we have monitoring to help make sure that all of these other elements are working properly. The last thing I wanted to mention is that none of this is static. This is undergoing constant evaluation and improvement all the time. And one of the processes that we've been evaluating is that related to the importation and rederivation of new mouse models. As you're all aware, introducing new mouse strains carry significant risk. We import hundreds of mouse strains from all over the world every year, and those are coming from facilities at pretty much any health status that you can imagine. And so as a consequence of that, we've had a strict rederivation policy in place for many years. And that's protected us from the agents that we exclude, but I think it's also protected us from newly emerging ones that we don't know to test for yet. And it's a process that to date hasn't failed. We have yet to identify any case in which we have seen vertical transmission of any agent that we exclude. The process that we use today relies on embryo transfer, and when it comes to preventing pathogen transmission, there are a couple of key components that I'd like to mention. The first is embryo washing. And in this process, we are just picking up the embryos and moving them from one batch of media to a new batch of media. And this is repeated 10 times for each group of embryos with a 100-fold dilution at each step and with a fresh pipette tip at each step to avoid cross-contamination. And this isn't something that we developed. This is an international standard that was developed by the International Embryo Transfer Society and is used not just with mice but with other species as well. And it's also recognized by other organizations such as the World Organization for Animal Health as really the gold standard for rederivation. The other key component is embryo recipient testing. After weaning, the embryo recipients are tested for excluded organisms. And this allows time for an immune response and antibody production because most of these methods are serologically based. As we've examined some of these processes, though, we realized that there were some risks inherent in the process. If you look at the timeline, you see that it's roughly about 7 weeks from the time that we do embryo transfer until the testing is complete and we can release animals. This means that there's a delay in detection. And during this time, an active infection could develop if there was a break in process somewhere, and there's at least a potential risk for cross-contamination to occur. And as I mentioned before, the process has never failed, so we know the risks are very low. But recognizing this risk, we sought out methods to try and reduce the potential risk associated with this process. And so what we've been working on over the last couple of years is looking at testing the embryo wash media. That is the final media that the embryos are bathed in prior to embryo transfer, as well as any leftover embryos. And we're testing the media using qPCR, testing for all of the 18 major pathogens of concern that we routinely exclude. So these are the same list of agents that you're used to seeing on our health reports. The method is highly sensitive with a detection range of 10 to 50 genome copies, and it's readily adaptable. So as we identify new agents in the future that we would like to exclude, it's going to be relatively easy and straightforward to add them to this testing system. It also enables very rapid reporting of results, and there's a growing body of literature that supports embryo wash media testing as being highly effective. So this method is it's effective, it's highly validated. We've been testing this over the last couple of years, validating it initially in spiking experiments to begin to evaluate the range of sensitivity. And then we've been running this process in parallel to our standard processes for the last several months to make sure that we have concordance in results. But to me, the real advantage of this is that it enables the identification of a potential break-in process prior to there being an active infection. So we're really moving to a model that will rely more on prevention rather than detection. And so this is really consistent with our focus on exclusion and prevention. One of the things that we've had questions about is what else is changing. What you'll see is that on the room in which we are housing the animals that are the embryo recipients, this is G200, there'll be a report, a section added to our health report that will describe the embryo wash media test results so that those are readily apparent. You will also notice that the number of animals tested in this room will begin to decrease some. Otherwise, operationally, nothing is really changing. The embryo wash process is unchanged. It's the same process that we've been using for between 10 and 20 years now, very successfully. And room operation is unchanged. This is a maximum barrier, so this is really our highest barrier facility at the Jackson Laboratory. All of those things that protect that barrier, including things like sterile supply, are going to remain unchanged. And we'll be ensuring the quality of the animals in this process through frequent testing. This space, or the animals from this space, are tested more frequently than pretty much any other room on campus, and that's going to remain the case, so that you can trust the quality and the health status of the animals that you're receiving from us. And so with that, I'll just summarize. The health monitoring for G200 is changing. This is a proactive change to improve health monitoring. There are no other operational changes that we anticipate. An additional section is going to be added to the health reports for G200 so that the health results are available to everyone. And with that, I'd like to thank you all for attending, and we can turn this back over to Janine.
Okay, thank you, Rob. That was a short and sweet presentation, but definitely is highlighting a lot of the important changes that are coming or that have been in place in our rederivation services. So I wanted to thank Rob for giving us a presentation, and I'd like to open up the floor for any kind of questions that you guys had throughout the presentation about what might be changing and what things are actually monitored. And there was one question that came in earlier about, presently, is there a list of organisms that are excluded, and where can we find those?
Yes, this is Jim Fahey talking. I can answer that question. We don't have a specific list of organisms that have come in with mice for rederivation that we have successfully eliminated. But what I can tell you is that we frequently get health reports with the mice that accompany the mice that come in for rederivation. And in those circumstances where those mice have been positive, for example, for mouse hepatitis virus, mouse parvovirus, minute virus of mice, and so on, we have successfully eliminated those pathogens.
Okay, great. And also, what are the typical pathogens that you actually monitor for? I think you mentioned about 18 organisms that you just routinely monitor for.
I think the best thing I could do is refer to people to the health reports that Jackson Laboratory publishes. And if you look at the front section of that health report, you'll see a list of organisms that we test principally by serology. A few are also done by PCR, and it's almost exclusively viruses, although we also test these embryos and embryo wash fluids for Helicobacter as well and Mycoplasma. But I suggest you look at that health report.
And that
Any
what
health
they're
reports
referring
in
to is
Jackson Lab,
actually
you'll see
from
that list.
every strain. You'll find this on the data sheet that there is a room that these strains come from, and every 6 weeks or so there's a update to those health reports, and so you can download those and see just what the most recent health report is, as well as the history from the last year. One question that's coming in is which passage are you testing the media?
Yeah, so the idea is that we are testing the media as well as the embryos that the embryo recipient would actually be exposed to.
Another question that just came in is, do you use embryo transfer also if the health report looks fine? I guess that's coming from individuals who are donating their strains in.
We use it no matter what. I think that's played a big role in protecting the health status of all the mice that we distribute. This strict rederivation policy has really, I think, been an essential element. I'll let Jim comment on it further, but we have a very strict rederivation policy.
Yeah, I agree with Rob. I think that has been the principal reason that we have never imported infectious diseases.
No matter what people say, we are going to rederive before bringing them into our facility.
Because of this strict policy.
And part of that is, even if the health report from the facility was fine, there's nothing present in the facility, we're aware of a number of anecdotal reports where it looks like the animals became contaminated during shipping. And so that was one of the reasons that we don't really trust what's on the
Great, yeah, because you don't know what could happen from one door to the next. Well, thank you all for attending, and I wanted to thank our speakers again, Rob Taft and Jim Fahey, for telling us, enlightening us about the processes for rederivation, and also for just the mouse donation and what happens when mice are donated to the Jackson Laboratory. I think these are a lot of important considerations, and it gives us a lot of information on what's happening as those mice are coming through the doors, and gives us a lot of security in knowing that there's a lot of rigorous testing, something that's obviously been done from an international perspective, but also this is a really new method that is not standardized and is actually going above and beyond what is normally done internationally.